Thank you. It's such a pleasure to be here. And now welcome all to this introductory session. This will be divided into two parts. For the first half hour or so, I'm going to interview the laureates to find out a little bit about their life and work. And then the second half of the session will be devoted to your questions. In fact, you can start submitting those questions now using the dialog box on the right-hand side of your screen. And I do encourage you to send your questions in throughout the conversation, please. But now let's meet the laureates, Liz Blackburn, Bob Grubbs, and Andy Fire. I think we'll begin by having you talk about your work a little, and let's do it in ascending date order of Nobel Prize. So, Bob Grubbs, you were awarded the Chemistry Prize in 2005, so you have the honor of going first. You were awarded the prize for the development of a new method in organic synthesis, metathesis. Could you tell us very briefly what it was about?
Yes, thanks. Yeah, and so I'm an organic chemist, and organic chemists like to make molecules which involve making and breaking bonds. And metathesis provides a really new way of doing that. That allows you to make molecules that range from pharmaceuticals to plastics and polymers. And so we developed this catalyst. I've studied it my whole career for 50 years, and in the early 90s, we made our first catalyst, and we've been making them better and better since then. But essentially, based on this new way of constructing bonds, it's resulted in, for example, the first hepatitis C drug had a component that was made using chemistry we had developed. There's 40 miles of pipe in the bottom of the Gulf that's is coated with a plastic that's made using the catalyst, and the largest refinery in the world that uses seed oil is going in Indonesia using the catalyst. And so, basically, finding a fundamental way of putting carbon-carbon bonds, breaking them and putting them back together again in new ways, opened up lots of possibilities.
Thank you very much indeed. Yeah. Okay, next to Andy Fire, who was awarded the Medicine Prize in 2006 for finding a new way of silencing genes, RNA interference. Andy, tell us about it.
So I was one of many people who were working on situations when genes would suddenly turn themselves off for various reasons. And often we didn't understand why. And sometimes we knew that we had intervened in the system by putting in a little bit of DNA or a little bit of RNA. And what I and many others in the field did was to try and figure out what kinds of things triggered that event. And my colleague Craig Mello and I and some of our other colleagues at that time, University of Massachusetts and Carnegie Institution, actually figured out that if you had both strands of RNA, for some reason, this gene silencing event was very, very efficient. And we named that RNAi after Craig did an online poll, actually. And that's essentially one of the ways that we have now of sort of doing the opposite of what Bob was talking about. We break bonds in a specific way. We break bonds in an RNA that we can target using the sequence of that RNA. And if you do that with just the right sequence and just the right delivery method, you can actually make change in a specific way in biological systems, which is what RNAi has been used for.
Thank you very much indeed. Beautifully succinct. Thank you. And Liz Blackburn, you were awarded the Medicine Prize in 2009 for your work on telomeres, the caps at the ends of chromosomes, and discovering this enzyme telomerase. Please tell us about that.
Well, there was a mystery in DNA, which was that nobody knew how the very ends of DNA were replicated. And there was another mystery. Nobody knew what the DNA at the end of chromosomes was. And so with a fortuitous system that allowed me to analyze the molecular nature of chromosome ends, actually by using very small chromosomes in a single-celled organism, I was able to find that it was a very unusual kind of DNA sequence. And particularly strange was that the number of nucleotides at the ends of DNA molecules, the ends of chromosomes, were changing. And so, long story short, we found that part of that change involves adding DNA to the ends of chromosomes by a then unknown enzyme that we later called telomerase. So I see a bit of a theme here about bonds. This is an enzyme that makes bonds. It adds DNA to the ends of the long DNA molecules of chromosomes. And how that all played out was the work of a great many people in terms of what that meant in terms of how our chromosomes are maintained during our long lives as humans. And essentially, the DNA replication process makes chromosomes more likely to shorten over time than you would hope. And it does happen, and telomerase adds back DNA. And we now know that we have to have intact caps at the ends of chromosomes with enough of this particular telomeric DNA in order to keep the chromosomes stable. And all sorts of things happen to our health and our disease processes if this is not a process kept in the right balance. So we have to make sure our chromosome caps are good and stable.
Thank you all very much indeed. When I was thinking about getting you all to describe your work, I was thinking, how am I going to bring you all together with these disparate pieces of work? And you've already done it you've done the job for me by bringing up this bonding idea.
We bonded over this,
We bonded, okay.
Yes, right. It's all chemistry.
Beautiful. Even online it works. Even virtually. Isn't that nice? Yep. It's the power of science to unite. So people will get the chance over the next 2 days to ask you lots of questions about these things, which is lovely. But the one thing I was thinking that probably does bring you all together is your motivation for doing this work in the first place. The results of your work, the applications are many and various, but why you actually started to pursue these questions. Was it with application in mind, or was it something else that drove you?
I mean, for me, I grew up in a rural environment and went to college to be an ag major. And fortunately, got to learn a little bit about chemistry. And then I discovered organic chemistry and was just fascinated by the intellectual component of it, which is this construct that's been put together that allows us to understand many different reactions and make predictions. But also there was a manual aspect of it too, which was going in the lab and making things and making new stuff. So the original thing for me was this combination of which fit my background, which was understanding how one thinks about molecules, how you make predictions, and then also how you go in the lab and then translate that into something. And the other thing too, which I laugh about sometimes, I was a really bad student. It was the first thing I found I was really good at.
Well, you were really good at it.
Yeah, okay.
Andy.
I mean, it's not totally that I just like to break things, but it can be fun if you are doing it in a guided way that's interesting. So, but I think looking at pattern and specificity was always something that was beautiful to me in biology, and being able to change things in a directed way was always of interest as an intellectual exercise and something to actually a tool to understand biology. At the same time, it was clear to me that the most transformative science came from experiments that were a little bit off the wall, a little bit unusual. And so I've always tried to do some of those experiments, and many of them haven't worked, and many of them they haven't worked because I did the wrong experiment or wasn't doing the right thing. But I certainly enjoy being able, when I can, to go into the lab or onto a computer and try something, ask a question, and get an answer that I don't expect. And that's what I enjoy about biology when it tells me I'm wrong.
When did you get the confidence to do that, to play and not mind being wrong?
It's more that I was fortunate to work in environments where that was supported as even starting with my undergraduate work and at a department where Liz was a faculty member actually at the time, and then moving forward as a graduate student and a postdoc in communities where that was a respected part of life. I hope we still respect that in science. Sometimes it's hard to justify giving resources to someone who not only might be wrong, but tells you, I don't know if this is right. I'd like to try it. Can I have the resources to, like, my time for the next 2 weeks to try this? That's a valuable position to be in, and I respect the people and institutions that try and make that kind of environment happen.
Thank you. Liz, please.
At the time I started the work on chromosome ends, really very little was known about DNA sequences, and certainly nothing was known about what was at the ends of chromosomes. So it was really a question of curiosity. I just realized that there were some chemical and enzymatic tools available that had just been developed as techniques for the very earliest ways of sequencing DNA, laughably crude and simple as we look at DNA sequencing now. But it was now it was possible to ask a question that nobody had had any ability to ask before. What is at the ends of DNA molecules that are naturally occurring inside cells and not bacterial cells and not viruses? And so I just wanted to know what was there. And then it just kept going because what was there kept on breaking rules. And I love the mystery of it, and I love the excitement that you didn't know what you were going to find. And I think that's one of the best things, as Andy is saying, about basic science, that the real joy is when you find things that you didn't expect to find. And so your curiosity and imagination get greatly piqued by these novel kinds of things that are showing up and are telling you this is what life is doing, this is what biology is doing. Let's try and find out why. Certainly it was with no thought of any particular application in human health or disease processes. It was just, let's try and find out. So that setting where the National Institutes of Health just said, go for the science, try and find out what you can, was wonderful and really enables science to happen in ways that you can never predict.
Thank you. Bob, please.
Yeah, I mean, just to comment on that, when I tell my students, if you run a reaction and you get what you expect, you haven't learned anything. But if you get the unexpected, then you really learn stuff.
I'm interested in this confidence thing because, in a way, I mean, Nobel laureates often point out that everybody starts out curious, and they're all driven by curiosity when it comes to it. But curiosity can sometimes be a little bit ironed out by school, studying for exams and needing to know the right thing to get the right grade and all of that. Do you find that when you have students coming to your labs that they can sort of relearn curiosity, or are the good ones who are going to do well already so curious that it's just obvious that they've got this insatiable desire to know?
Well, I could comment on that. I don't think they all come in with a one-size-fits-all, I'm curious. I think that some come in with very different motivations, and having an environment of training where you can say, it's important that you explore and you develop your own ideas, and a lot of science will be failure. I think one of my most revealing moments was I looked up in my notebooks, because I was asked when I got the Nobel Prize, when I look up in the notebooks and I go through my notebooks and I find that 90% of the things probably I tried didn't work. And so somehow our memories are enabling us to be very resilient to what I can call, what seemed like failures at the time, which is that you sort of just keep going with the ones that do lead you down productive paths as much as you can. But a lot of ability to, quote, fail, I think is very important. Otherwise, if you're not failing, you're not pushing boundaries. So I think we need to understand that basic science looks inefficient from the outside, but it's really important to be able to just push the boundaries. And sometimes a failure later on, you say, aha, now that feeds into this question or this kind of experimental approach in ways that you didn't expect. So I think this confidence issue is, you have to have people understand that it's really okay to keep pushing, so long as you're thinking well and thinking rigorously and doing it as well as you can. That kind of failure, I think, is what is part and parcel of pushing boundaries.
Thank you.
I feel like I came up in a good time when there was sufficient funding so you could fail. I'm hoping now with the pressures on funding and the cutting of funding that one has to be much more certain, and there's much more tolerance of failure. I mean, much less tolerance of failure. Yeah.
That's interesting. Andy was also hinting on a possible change in the environment now. Let's turn to the environment. I mean, another thing that unites you is that you've all chosen to live in California. I was reading an obituary for the statesman George Shultz the other day, and apparently he was at Stanford, and apparently when ex-president of Russia Mikhail Gorbachev visited him, he said, "Ah, George, I see you have chosen to live in paradise." So here you all are in California, living in paradise. But how important is it to find yourself in a place that is really sort of well funded and everything, has all the advantages, or can you do great science without being in that sort of place?
My contributions in RNAi were, I was born in California. I love California. It is not paradise. Anybody who has dealt with our fires in the last few years, our politics, anything is going to know this. One can do science in a lot of different environments. The people are the most important component of it. And that's the environment. That's really the environment as it's defined. Money can be the end of things. If there's too much money and too much responsibility to account for it, it actually becomes a detriment to doing good science. And so the groups can get too large, the problems can get too diluted. And so there really is an advantage in working in a small environment where what is the best thing for me is to be able to come and think about a problem, tinker with it. You do need tools for it. The tools do, in some cases, cost money. You need colleagues to bounce ideas off who are going to be accepting of really crazy ideas, but also be critical and try and improve them. And that can come in a lot of different places in a lot of different forms. And that's why you see people from different environments. California is a place where a lot of scientists have converged, but other places as well. And you can see a lot of really great science being done throughout the world in different environments. So I would not say California has anything unique. Everybody come visit. Before you decide to live here, spend a fall here with the fires happening.
It's really people. And, when I think about the practice of science, if you will, over the years, the thing that stands out is just the wonderfulness of talking about science with colleagues and bouncing ideas off, and just having that process of human minds coming up with, more and more interesting kinds of things that honestly by yourself you wouldn't have dared necessarily to do, or you wouldn't have thought it through properly. And so, some of the really good science is done because you have to think in elegant ways, because you don't have infinite resources. And so you have to think much more smart about it. And that can make you into a good scientist. But it's certainly not unique to California. And over the years, I remember being struck by the fact that, I'd go and give a research talk, a lecture at various places around the country. You'd be invited to talk, and then you'd spend a day or two talking with scientists there. And first of all, I was thinking, why am I getting on a plane and going to this place and talking? And oh gosh, I could be back at home in the lab or with my family or something. And then you'd realize every place you went, no matter how sort of seemingly obscure, you would have had very likely at least one totally unexpected conversation about science with somebody you'd never met before, whose research you never knew, and you realized there were just tremendously smart people everywhere. And so I like to sort of have this image of really good science just bubbles up everywhere if you let it bubble up. And so I wouldn't be at all restricted by saying, oh, it's California. There are places everywhere, no matter how small and obscure, where it's the minds and the thinking of people that really just shine.
Bob, please.
Yeah. I mean, I moved to Caltech for the students and the people, and it could have been any place. I'm certainly happy it's in California, so I can go walk and enjoy the outdoors and fish and do the other things. But it's really down deep. It's the people. And so recently I had a student who had 2 job offers. One was a very good salary, lots of startups, and the other one, it was at a phenomenal place that had really, really good students. And my advice, which I think he takes, is that you go for the students. The money runs out soon. It's the students, and it's the quality of students you work with. And having the ability to sit down in an office and starting talking about one problem and ending up on another one, and people are willing to take new chances. I've changed, I've gone into a lot of different fields, and it's because the students are willing to do that. And sometimes I tell them their hardest job is going to be teach me.
Now we're on the subject of talking and sharing your science. I suppose as a young person, it's often a little bit difficult to know how much you should tell people about what you're doing and how much you should keep to yourself in case somebody takes your idea. What's your advice? Should you just always talk? Andy?
I mean, I had a wonderful graduate advisor who used to say that, it was much worse to be ignored than to be scooped. There are times when an idea is more ready to talk about in one environment than another environment, but there's rarely an idea that's so unique and so perfect that it wouldn't benefit from feedback from people. So it's usually more a matter of trying to find the people that can give the best input on something rather than to try and hide it from somebody. You're trying to get it to the most useful person to be able to give you feedback on it over time.
I heard it once said that in business you try and kill your competitor, and in research you actually want your competitors to thrive, because that's making even better science happen. And I think there was some understanding about this in the computation software industries, where when things were being developed very early, they were open source because people realized things would move faster and better. And so I've always felt, yes, you should share your ideas. You might get scooped, but as I would tell my students, who more or less believe me, I'd say, look, what will be remembered is the good paper. That's the paper that will prevail, not necessarily the one that came out, a few months earlier or something. And so I've always felt it's always been so much more rewarding. I mean, I just talk to people. Maybe I'm just a blabbermouth and I can't stop, but but I'm excited. I want to talk with people, and it pays because you get such interesting input. And biology, luckily, is so complex that even if a couple of papers published, on a similar subject look as if they might be the same, there are always nuances that are different because there's such a richness and complexity in biology. So I really think that openness in science, all the way to open access publishing and everything that we do, it benefits enormously by being open.
Bob?
I like to babble and like to talk about science, and so I mostly do it. Unfortunately, in every field, there's a few people who are not trustworthy, and so those you're a little more reserved with. But when you have your friends and you develop your group of colleagues who you trust and you work with, and so that's really what makes it all worthwhile, is having that group that you can trust and talk and be totally open with.
Thank you all very much indeed. Okay, so there's just a few minutes left, so I just encourage the audience, please do ask your questions using the dialogue box, just before we go into the Q&A. But you've hinted that things might be a little different nowadays, partly the funding environment, making it harder to play around, perhaps. But what do you think? Are there fundamental differences between when you were growing up as a scientist and now? Or is it basically the same? Liz, what do you think?
Well, it's hard to put yourself in the shoes of somebody who's growing up in science now. Although when I look at the experiences of the trainees and the students around me at the University of California, San Francisco, I try to envisage their world, and I think at some level it's really the same. I mean, people are just doing research because they find it extremely interesting, and even though there is this sort of push towards appliedness. The lesson I had once was when I had an MD PhD student, and so I gave him the project that I thought would be dearest to his MD part of his heart, and he immediately chose a project in my lab which was the most fundamental kind of basic science one. And so I thought, I'm never going to pigeonhole people who are, I would have thought would be wanting to do applied things into that kind of thing. I think that I see the motivations of students as they just want to know. Some people think more in terms of their careers because they do think more strategically than perhaps we did when I was training. But the fundamental, just loving to talk about science or do science, make discoveries, that hasn't really changed. The outside environment, sure, has changed. Digitization, access, the way you can approach things with different technologies, absolutely. Those have made terrific and very exciting changes.
And indeed, we'll be having a whole panel discussion around the idea of how technology and digitization is changing things. Thank you. Andy?
I mean, maybe I would beg to differ a little bit, or maybe accentuate a different point than what Liz just said. I think things have changed, and it's really come into focus, not necessarily just in the last year, but in the last year, in that science is such an integral part of the human race managing to sustain itself. It was always true. I mean, we needed medicine, we needed antibiotics. But I think that the turnaround time is much more so at this point, that we really need we're going to need biotech to generate food. We need to have ways of tracking infections. We need to have ways of dealing with infections and preventing them. We need to have ways of dealing with the environment. A lot of what we're dealing with is what humankind has done, but also a lot of what we're dealing with is that change is really fundamental, is going to happen. And at the present, the way of having several billion people and dealing with that is science. And so it's a mixture of the great need that society has and the great fun and joy that science is. And that's a different equation. And that's where, biotech and academic and traditional industry and government are all areas where science is going to be critical. And I don't think we've actually begun to understand the degree to which we'll need people that are scientifically trained in all of those areas. And so I think it's a little bit of a different world. Not that science wasn't important before, but I think the immediacy of scientific discovery being needed on a short term to deal with sustainability issues is something that we're going to be understanding in the next few decades.
Thank you very much. And indeed, we'll be having a panel discussion that will touch on that as the closing panel discussion of all this on the value of science. Thank you. Thank you. Okay, Bob, last word to you.
My early days has changed. So, I grew up right after Sputnik, whenever money was flowing into science, and, everything was fundamental. As we moved along, I think there's much more emphasis now on having things that at least tie somehow to practicality. For example, the National Science Foundation, now you have to write a statement on broader impacts. So it's, how is this going to influence the field, and how is it going to influence the future of things? So I think part of the thing, as you said, we need more science, and I think more and more there's going to be more and more pressures to go into things that are fundamental, but have an outcome that could really have an effect on the society.
Thank you very much. Sorry, Andy, did you want to come in?
Yeah.
No, I agree with all things, and I fundamental science is really core to a lot of what we're doing. But there's also, I think there'll be a shift in a responsibility that scientists have to build their own structures to make the science work for society.
So I look forward to continuing this. But for now, it's time to go to your questions. And so after a momentary break, we will move into the Q&A. Thank you all very much. Liz Blackburn, Andy Farr, Bob Grubbs. So welcome to our live Q&A. And I'm very pleased to say the questions are already flooding in. So let's start straight off with a pretty fundamental question from Sophia, who asks whether you have a role model yourselves. Liz, what about you? Do you have a role model?
I think I do. Two. One who I never met, and that was Marie Curie, the scientist who discovered radium, among other things. And I read a book about her when I was a child, and just was very impressed by how much she loved science and many of the aspects about her. But in real life, I also have my role model for what I have done in science has been my postdoctoral advisor, Dr. Joseph Gall, and he was just a wonderful postdoc advisor in many levels, from scientific to being very supportive and kind, to his trainees. and to also just how you conduct yourself in science. So I felt very lucky to have had one I know in real life and one who I met through a book.
Thank you. Andy, what about you?
I've had a lot of role models from my advisors at various stages. Hung-Chi Wu as an undergraduate, Phil Sharp as a graduate student, Don Brown as a young faculty member, Steve Galley as a slightly less young faculty member. Also, my students, in some ways, are my role models too these days. Just watching, really impressed and inspired by the way they've handled the last year in particular.
Bob?
Yeah, I was thinking, there hasn't been one. I've been through sort of many phases in my life, and sort of each one I've been lucky to have a role model for that phase who has done things. The person who really sort of changed things the most, who was the one who introduced me to chemistry and provided a model for really working in science and doing things, is named Merle Batiste, who was my undergraduate advisor and master's advisor. A PhD advisor for a while, although we were from such different backgrounds, it was difficult for us to have that role model thing. And since then, I think since then it's been just a number of people who at various stages. There was a senior faculty member who moved to Caltech, who became sort of the model for how I operated at the time, and sort of a great advisor, who's Nelson Leonard, who was a really fine gentleman. So it's been multiple ones.
Thank you. Do you think it's important for people to find one? I mean, you all have them. Do you think it's a necessary thing to find someone you want to emulate in some way? Who wants to tackle that?
Well, I think you'll have your own vision, and if a role model is somebody who helps you make decisions or makes you aspire to something, then that's helpful. But I think some people are internally driven enough that they don't necessarily need to have a role model. It's more something that makes you feel, this might be possible, or this is a kind of way of going about things that I want to be like. And but I wouldn't say necessarily, because you may have your own view of things. So you'll know when you need a role model.
Walter Romanko asks how much of your success comes from inspiration and how much from hard work, which is also a way of saying how hard do you really have to work to be successful. So, Andy?
If I really worked hard, the question could be rephrased: if you really worked hard, could you have done more? I think there's a combination of luck and perseverance. Hard work is useful when you get excited by things, and finding something that you can work hard at and you're excited by is a part of the whole deal. I'm not sure that just hard work is the key to being successful in science. I think it has to be a lot of introspection and thinking about what the right thing to work on is, and then once you find that, being inspired by it.
What about you, Liz?
I totally agree. I think it's such an interesting mixture of both inspired thinking in some ways, and then you just have to really kind of roll your sleeves up and do it, and then also know when to stop. That's a hard part too, and that's where really some of the very special aspects of being a scientist, I think, come in, because of this this kind of whole interesting, I think Andy said, intersection of those different qualities. But yeah, you have to work hard, but it comes in phases too. There are times when you just have to be working, especially in biological sciences, the demands are that you often do have to spend very long hours just because of the way the biology is working. But that won't be forever. So it's not like continuous drudgery. It's more you're rising to the occasion to meet what that set of needs is to get certain things done, and then you would kind of have your own time. And I've always thought it's very important to have time where you go away and do things completely different and not be a drudge 7 days a week, every hour of the day and night.
Yeah, all things are relative. I guess it's how much of your time is free time. Bob, and how long are the hours in the lab you really have to put in?
Yeah, I mean, there's some people who when I was a graduate student, I spent an incredible number of hours in the lab. As you move through your career, you spend fewer and fewer time in the lab and more time doing other things. But I think the key is to have time to think about stuff. I mean, I've had postdocs who come in my lab who basically work for someone who they work 24 hours a day for 5 years and published massive amounts of papers. I feel like my job as a postdoc is to let them have time to think and do things and sort of change their career. And one of the people recently said, the main thing I remember about you is coming through and saying, are you having fun today?
Yeah.
And so if working really hard is fun, and sometimes it is, then it's great. But if you're working hard just to work hard and for other reasons, then it's not so.
Okay, thank you very much indeed. Now, Sriranjani Daba wants to know how you come up with your best ideas, or also, I suppose, where you come up with your best ideas. Where does inspiration strike? Who wants to jump in on that one?
A lot of inspiration strikes when you're talking to somebody and about how things might work, how they could work. And that's why meetings are very important. Sometimes they're formal discussions and listening to a seminar, but sometimes they're just conversations. What do you think about that data? What should we do about this? And then things move from there. So I think communication between people is a key part of it. Sometimes you do get an idea walking by yourself in a pristine forest with nothing around, but that's somewhat the exception rather than the rule.
I get inspiration. It's almost embarrassing because I often found that when I was listening to a scientific talk at a conference and someone was presenting really interesting things and I'd be taking notes, but part of my mind would be also thinking about my own research, and some of my ideas have really come when I've been listening to someone's talk on something different, ostensibly, and I've had ideas that then have been useful. So I'm embarrassed because it sounds like I wasn't listening to the person who was giving the talk, but I was. So it's a very curious process. I think your brain just must get stimulated when you're listening to somebody talking, and suddenly other ideas pop in.
So, yeah, sounds like you were processing what you were listening to. Yeah, absolutely.
Or something.
Bob, where do you?
Daydreaming. No.
Well, daydreaming is pretty important, right? I mean,
absolutely.
Exactly.
Can't emphasize it enough. I enjoy telling parents of younger children. I say, they say, "What's important?" I say, "Daydreaming," and they just look at me horrified.
Yeah, right. Yeah.
There should be periods in school set aside for daydreaming.
Daydreaming.
Yeah, seriously.
Yeah.
Bob, where do your ideas come from?
Yeah, they come from different places. A lot of the best ones come when there's an unexpected result in the lab. When the student's doing experiments and they expect one result, they get another one, and then suddenly that sort of opens up a whole new range of things to think about and to do. And that's one source. The other one is, I was talking. I talk to people in different fields who come up with interesting problems in different fields, and trying to put their problem together with my past experiences and all those things. So that's resulted in some really interesting areas and things to think about. So it turns out there's lots of sources, and a lot of times, your best ideas turn out to be mistakes. You wanted one thing and you end up getting something else, and so suddenly that looks like a brilliant idea. It was just a mistake.
That leads beautifully into two questions which have come up, one from John and one from Camilla Ugaz, who they ask, how do you decide which ideas are going to be fruitful and which ideas are not going to be fruitful? Or put another way, the way that Camilla put it, is how do you decide which failure is worth thinking about and which failure is not worth thinking about? So, Andy?
I mean, that's a great way to describe a question, and it is that way. I mean, there's a little bit of, you think about what you can do in a straightforward way, and that does inform what you actually do. An experiment is going to take 2 years, then you think about it carefully. If you can do it in an afternoon, there's a low cost to that. You just do it.
Yep.
If there's a collaborative experiment that each person just has to put a little bit into and you can get an answer, that's another great place to be at. But it's a complicated decision, and in the end, you sort of moisten your finger and put it in the wind and see which way the wind is blowing, and that's a little bit of how you decide what to do.
Bob?
Yeah, it's it's very similar. It's if an idea comes up, it's a question of how long it's going to take. And many times ideas in my area of chemistry we can do rather rapidly. And so if it's an idea a student has and they want to do it and it's safe and it's not going to take very long, we always have to worry about safety in chemistry, and it's not going to take very long. And there's actually some useful things that will come out of it. Then you should go try it. But as you say, if it's going to take 2 years and going to be dangerous and you're going to have to make a big commitment, then that really takes a lot of time to think about and decide what the outcome is going to be and what the probability of success is, et cetera. So that makes it a much more complicated situation.
But how do you know what sort of question to tackle? I mean, I suppose some questions are insurmountably difficult or strike one as being that, and some questions strike one as being possible to answer, and some questions strike one as being a bit easy to answer. Is there a general guiding rule for students, people embarking in research? What sort of questions should you be tackling, Liz?
I think if you can design some experimental way of really rigorously testing it, then that to me is a really helpful criterion, because no matter how important or not important sounding it is, and that criterion is very subjective, what's important, what's not, because you never know. But if you can design something that really sort of nails, yes, this provides very concrete, really good evidence for, or conversely, really undercuts and says, no, this is very unlikely to have stood the test of experimental, rigorous in examination, then I think that's a great guide, because that makes something that really adds real knowledge one way or the other. So it comes down to me is, what is the kind of experiment that you could do that would be just terrific experimentation? And so that comes back to the inspiration, what not inspiration, but how do you decide what to ask? I think always subjected to what can you subject it to, and it's a matter of, is it doable or not? Sometimes there aren't the technologies to ask certain questions.
Andy, do you want to have a crack at that one as well?
Yeah, I mean, I guess I would just agree with Liz. I mean, sometimes what drives you is really wanting to answer a question and not having the technologies, and then you have to decide, who can you go to get that, or if all else fail, how can you develop the technologies to do that? And then that's another thread that a lot of our science has is developing technologies to answer questions. The barriers that the technologies face are often biological barriers in our world, and overcoming them means sometimes understanding the biology even more, and so that can be a conduit to actually learning things, of trying to develop technologies. So there's a synergistic advantage to developing technologies in biology and understanding how and when they work and when they don't work.
Thank you. Okay, we're moving into the realm of now kind of mindsets. So Adam Christensen has asked what sort of mindset a student should have to prepare themselves for going into a research career? I'm not sure. That's quite a big question. Does anybody feel able to tackle it? What's the right mindset for a researcher?
I'm not sure there's a wrong mindset for a research career, but it does, if you're having, as Bob described it, if you're having fun doing research, then looking for an area where you enjoy the research and where it benefits humanity, and therefore hopefully is a job that somebody will pay for. Those are good criterion. The mindset, I think, does change also from day to day.
Yeah, that's true. That's true. Well, that actually leads us to a second question on the same sort of thing. And this may not be a problem that any of you suffer from, but it's from Clark. And I suspect Clark might be a researcher whose experiments aren't working, because he asks, how do you cultivate your curiosity when it runs out? Bob.
What?
Yeah.
Sorry, Liz, go ahead.
Go ahead. Yeah.
Liz, you go first.
Well, I think that might come from you're feeling burned out or something, because curiosity is such a part of just not being a scientist alone, just being human is, and so it might be coming to something that is just you're exhausting yourself. So I'm coming back to this point I said earlier, which is that not only it's not just hard work, you also have to really have a break to kind of refresh and recharge yourself. When I was junior assistant professor faculty member at the University of California, Berkeley, every Saturday I was exhausted. And so I would just sort of do completely other things and relax. And then by Sunday I was fine again. So you just have to keep something that keeps your zest going. Now, if your curiosity is running out because you really aren't interested in the work, as was said, maybe if you're not having fun, then that's pretty, at least some of the time, then that's pretty indicative that maybe this is something that you don't really want to be doing. But I would say take a break and recharge first and see if the curiosity comes back.
Has it ever occurred to you, Liz, that you wanted to do something else? Or have you always been just content being a researcher? Liz?
I thought the most wonderful thing in the world would be to be a classic concert pianist. Luckily, I realized I had none of the talent required for this. So I kind of chose science as, okay, I could be okay at science and not have to, because if you're going to be a concert pianist, you would have to be absolutely superb at it. But I thought there are more ranges in science to be participating in science and still, it would still be okay if you weren't absolutely marvelous at it. You would still find it interesting and fulfilling, in some ways. So that was my teenage thought. Luckily, I had a little dose of reality.
Thank you. So I remind people we've got 10 minutes left, so do submit your questions if you want to ask the laureates anything at all. Everything's fair game. Coming back to the question of failures, Calla asks whether it's a problem that failures tend not to be published. Bob, what do you think of that?
Yeah, failures that's always claimed my whole career is based on failures. And so basically we try to do something and something else happens and then we learn things and that puts us in the right direction. And I've, in many of the graduate students I've known who have been, incredibly smart and ambitious, make really poor researchers because they can't deal with failures. They think about something and plan an experiment and they can't deal with the psychological problem of what they're thinking not working and being wrong. And so I think dealing with failure is really a key piece of being able to do it. So not only do failures sort of put you off in a new research direction and a new way of doing things, but also you just got to learn to deal with them and keep going.
But do you think there's more of a role for the journals in publishing failures? It's difficult, but does Andy, what do you think?
I mean, it's useful to do, but I would point out another aspect of it, which is that one of the wonderful things about being in academic science is if you do an experiment and it doesn't work, you actually cannot do it again. You can just go on and do something else. You don't have to be absolutely sure that that idea is a bad idea. And that really does sustain us to some extent, is that you can go on to the next thing and you can look for things that do work. And so to be really sure that something is a failure can be a ton of work. To be sure it didn't work the first, second, and maybe even the third time you did it may not be as much work. So that's something to be, and as Liz said, it's important to know when to give up. And sometimes you don't give up for a very long time. And sometimes that was a cool idea. I'm going to do something else tomorrow or next week or next year. So it is there is an advantage in not pursuing every failed experiment and not even needing to tell anybody that you're absolutely sure it didn't work, because you're not sure, it just didn't work for you.
What
Yeah.
about
And
the
I think there's different
Yeah,
kinds of
please.
failures. There's failures because something experimentally didn't work, because it was technically too challenging, or you messed up or something like that. And then there's failure because you define it as failure because your favorite idea, it didn't work out that way. But if you can feel really sure that the experiments were sound and really were well done, then you can kind of turn that failure into, well, I've learned something, and that can become publishable, though you don't label it failure. But you have to also have really tested, did your experimental whole approach and so forth, was it working? Did you make sure that it was not just because of some silly screw up? And so you have to think of failure in different ways. But I agree. I think 90% of what I've done, I would say probably when I look in my notebooks, lots of things don't work either the first time or because it was just too hard or it was the wrong idea. So it's a very strange psychology. We have to live with failure all the time and yet somehow be very resilient and say, it's okay. And I'm prepared to give up my favorite idea because the evidence is just not going in that direction. So resilience is I think a surprisingly large amount of what we as scientists have to do, but we kind of hide it well.
Yeah, the other part of it is about publications. So and so it would be good to know if an experiment's been done before and failed, but that doesn't get published. But it's changing now in chemistry in particular, and maybe in other fields, is that people are starting to apply artificial intelligence to chemical reactions, and under those circumstances, what failed is just as important as what worked. And that data is really not there. So I think there's going to be a lot of efforts now to start to look at a reaction over a whole range and show the bad results and the good results, so it can be plugged into to programs that then start to make predictions.
Okay. Thank you both very much indeed. I mean, sorry, all three of you. I was thinking both I was thinking of both the questions I'm going to try and treat in one go here. That there's a question about mentorship. What's your mentorship style? But then also a question that's from Calla again. And then there's a question from Macy, who wants to know how you cope with having too many research ideas, or rather, if you had a student with too many research ideas, what would you advise them? So how do you look after your students? Briefly. Liz.
Well, I've had great good fortune. The students who I've worked with, and these are mostly graduate students, but sometimes undergraduates, have really wanted to be engaged in science. So motivating them is no problem. I think it's often showing people that they have potential that they may not have had. And this is true often for the women students, that they don't have enough confidence about what their abilities are. And on the other hand, you also have to be honest with people, and if I think they can see that you care about their career and their success and so forth, and you really don't think they're suited to science, then I think you can deliver that news. And I've had to do that a few times, and it's hard, but I think you need to be approaching your students, and they're part of your sort of extended family in a way, and so you need to be supporting them and also saying when things are screwed up. And being a little bit tough sometimes. Once somebody in the lab once said, in our lab, we think Liz's motto is no whining. And I was kind of shocked because I thought I was very kind. But I think you have to have that sort of mixture of tough love but fundamentally really convey that it's important to you how your trainees and colleagues are thriving.
Thank you. It's nice advice. Questions are pouring in now. We don't have very long, and I think we should finish on something fun and revelatory. Mark Erat wants to know whether being awarded the Nobel Prize has changed the way people treat you. So do people treat you differently, Andy, now that you've got a Nobel Prize?
Well, my last grant rejection came about 25 minutes ago in the email. So, I mean, it doesn't really change life, and it shouldn't change your life. There shouldn't be any difference whether somebody's a Nobel Prize winner or a grad student. I don't treat the ideas that come any different from anybody, and nobody else should either.
Thank you.
I think within science, it hasn't changed things. Outside of science, people tend to believe you a bit more than they ought to. I think that's the big difference.
Bob?
I think you're right. Yeah, inside science, it hasn't changed very much. In terms of how things work and how you deal with things. So some people, make it that way, but I think from dealing with this group, I think that's not their style. But yeah, outside it's I get often asked if I won a Nobel Peace Prize, et cetera, and so there seems to be something associated with that. And and so in some circumstances it makes quite a difference. In some countries it makes more difference than others. In the U.S. on the Caltech campus we have lots of Nobel laureates, and so it doesn't make any difference.
So does it get you a parking space on the Caltech campus?
Well, everybody at Caltech has a parking space on campus.
Okay.
So.
It's
Southern
Berkeley where
California
parking spaces are hard to get. Yeah.
Yeah.
Well, the University of California system, while they give you a parking place, you still pay for it. And secondly, I remember getting a letter from the president of the university, and it said, "Congratulations, you are the sort of like blank space 60th or something like that. University of California person to get a Nobel Prize, but congratulations anyway." so
yeah,
doesn't make that
keeps
much
you humble. Thank you all very much indeed, and thank you for the lovely questions. It's been such a joy to read them, and I'm sorry we couldn't get through them all. They are piling up. It always happens. Lots come in at the end. But thank you very, very much. So now you'll meet the laureates again in their next panel discussion, which is on how digitization is changing science, and that's going to be screened at 1:30 Austin time. So do please return and tune in for that. For now, we'll say goodbye to our laureates, and if you want to stay tuned, 3M are going to take you on a tour of the 3M Austin Science Center and the Austin Innovation Center in a beautiful 15-minute video. So do stay tuned for that. But for now, thank you to the audience, and thank you very much indeed to our three laureates.